In this Chapter 15 video, we are going to be taking a look at ATP. So we're looking at this portion down here, the row of ATP, how it's used, and some examples. Okay, so we know that ATP is the energy currency of the cell. And ATP, it does have that phosphate group that's going to be transferred, and that's where we get that energy piece from. But ATP we use as our energy currency. There are other phosphate esters that exist that we don't use as the energy currency. So why is ATP better than some of these other phosphate-containing substances? And really that has to do with the free energy that's associated with the hydrolysis of the ATP versus the hydrolysis of other phosphoryl groups. So here, if we're looking at the hydrolysis of ATP, we see the delta G is negative 30.5 kilojoules per mole. If we look at another substance, and an example of that is this glycerol 3-phosphate, we see that the hydrolysis of glycerol 3-phosphate produces only negative 9.2 kilojoules per mole. So there's much more free energy associated with the ATP than there is some of these other phosphate substances. So ATP has a higher phosphoryl transfer potential than some of the other molecules, in this case, than glycerol 3-phosphate. And the higher transfer potential is going to be based on 4 factors. It's based on resonance stability, electrostatic repulsion, increase in entropy, and stabilization by hydration. So we can look at these all individually, and if we're looking at resonance stabilization, the orthophosphate is a product of ATP, and the orthophosphate has greater resonance stability than the other phosphoryl groups of ATP. So we're stabilizing the structure because of the rearrangement of those electrons and that partial double bond characteristic. In terms of electrostatic repulsion, at physiological pH, that triphosphate unit of ATP has about 4 negative charges associated with it. And those charges are going to repel one another. So here are the negative charges. They repel one another. And when we have hydrolysis of ATP, that repulsion is going to be reduced. And so, it's desired to have hydrolysis of the ATP. It minimizes the repulsive forces. In terms of increase in entropy, there's really not much more to say than the products of ATP hydrolysis increase the entropy because we had one thing and now it's broken into 2. So one molecule becomes 2. That's increasing entropy. Stabilization due to hydration. When we're looking at the structure, water will bind to the ADP and that phosphate, and it stabilizes these 2 molecules. So when we have water binding or interacting with these 2 substances, it makes the reverse reaction unfavorable. So we're not going to see the ADP recombining with that inorganic phosphate to produce ATP. This is not something that is a favorable process, and that's because water is going to help stabilize these products. So when we were talking about the phosphoryl transfer potential, this is important in terms of the cellular energy transformation. And there are a number of different substances that are going to have different phosphoryl transfer potentials. Some of them are higher than ATP, some of them are lower than ATP. So you see that there are different potentials associated with these. And based on the position of ATP, we see that it actually is intermediate in terms of its transfer potential with some of these things. So it lies at an intermediate position, and that allows it to efficiently function as a carrier of phosphoryl groups. And we'll see how some of these other things come into play and can transfer their phosphoryl group to ADP, and then the ATP can transfer its phosphoryl group to other substances. Here we're just looking at a table of some of the free energies of hydrolysis associated with those molecules. So this is the data that's associated with that previous figure. We can see the energy associated with the hydrolysis of those phosphoryl transfer substances. Now we can use creatine phosphate as an example for this process, this phosphoryl transfer. In terms of the creatine phosphate, free ATP is not going to be present in great enough concentrations in muscle tissue in order to sustain activity for more than a fraction of a second. So instead, we have these other molecules, such as creatine phosphate, that can serve as a reservoir of high potential phosphoryl groups. And these groups can be transferred to ADP in order to form ATP when ATP reserves are gone. And so we're just kind of seeing this reaction here. We have our creatine phosphate and our ADP. That creatine phosphate is going to transfer that phosphoryl group to ADP to produce the ATP, and creatine is left. So this figure is just showing that process. We have, we can see how ATP is rapidly, and this red line here is going to rapidly be depleted during exercise. After that point, the creatine phosphate is going to kick in, and it serves as a major source of phosphoryl transfer group to regenerate some ATP. But then that reserve is going to be depleted, and once the creatine phosphate reserve is depleted, then the ATP is going to have to be generated through metabolism, and that will happen through aerobic or anaerobic metabolism. So we have ATP that's present, quickly depleted, other high phosphoryl transfer potential substances like creatine phosphate in muscles can come into play and transfer a phosphate group to regenerate the ATP to continue to fuel the tissue. But then at some point that's depleted and we have to make more ATP. This is just looking at the ATP-ADP cycle. This is the fundamental mode of energy exchange in biological systems. The supply of ATP in the body at any point in time is limited, and the turnover rate is very high. So we have a quick turnover of this process. Having a process that allows us to easily, quickly regenerate ATP is going to be very important. This is the primary role of catabolic processes. So again, here we looked at the phosphoryl transfer potential of various substances and why ATP is going to be the currency that we use. And part of that is because of its intermediate position and can easily transfer and easily accept those phosphoryl groups.